Right, we'd better start. Welcome everyone. My name's David Delpy and it's my privilege today to introduce the speaker at this lunchtime lecture, Professor Clare Elwell. Clare's a professor of medical physics at UCL and she develops novel optical imaging and monitoring systems for looking at the human brain. Her research projects include studies of autism, brain injury, migraine, malaria, and I'm sure you'll hear lots of others from her today. She currently leads the BRIGHT project, Brain Imaging for Global Health. I think all projects have obviously got to have an acronym that is catchy. And that delivered the very first brain images, optical brain images of infants in Africa. She's president of the Society for Functional Near-Infrared Spectroscopy and of the London International Youth Science Forum. She's won many awards for her research, teaching and her public engagement, and she was a 2018 British Science Association Media Fellow at the Financial Times, and she's a founder and trustee of the charity Young Scientists for Africa. I'm sure we're going to really enjoy her lecture. Clare, over to you.
Thank you very much. Thank you, Dave. So thank you very much for all bringing your lunch here this afternoon. I'm going to start, I'm assuming that we have a very general audience here, so I'm going to start with what my definition is of the discipline that I've been working in for almost 30 years now. For me, medical physics and biomedical engineering is about addressing unmet clinical needs with innovative physics and engineering solutions. So as physicists and engineers, and I can see some of my colleagues in the audience here, our ideas are about not just thinking this would make a brilliant thing that we could build, engineers love to build and create things, but more to think where are the problems that we need to solve and to make sure that any of the innovations that we create are actually answering a question that needs to be answered. So I'm now going to move on to what I believe is a really hidden but very important unmet need. I spent a lot of my career developing technologies for use in high-resource settings. I've worked with colleagues in neurointensive care units, in premature baby units, thinking how can we make this technology even higher tech? And a few years ago, I was introduced to an unmet need that I was broadly, completely unaware of. And it's this statistic here. 1 in 3 of the world's children who are born in the poorest communities fail to reach their potential. Essentially, these children are surviving, but they're not necessarily thriving. And what do I mean by that? Well, I'm basing this on a paper that was published that's generated from lots of information from a range of studies showing that 1/3 of preschool-aged children living in low and middle-income countries are failing to meet their basic milestones in either their cognitive or socio-emotional development. This is 1/3 of children. This equates to over 80 million children. So when we look at the statistics of the number of children dying because of poverty in a range of different populations, this sort of statistics get hidden. These infants survive, but they don't necessarily thrive. And so what's the background to this? Why is this statistic so stark? Well, we need to think about the factors of early childhood adversity. What do we mean by this? Well, 1/2 of the world's children in the world are living in poverty, and they're exposed to a range of different risk factors. These include getting not enough food, but also getting food that's not necessarily delivering the right balance of nutrients for them. They're exposed to a range of infectious and non-infectious diseases. They don't have adequate health care all the time, nor do they have adequate provision of early years education. And all of these things have a particular impact on the growing brain, particularly because from conception, from conception to their second birthday, there's the most rapid cascade of development and maturation of the brain than any other time in their life. And so if these factors hit them very early, they can have devastating consequences for the remainder of their lives, not just in terms of their potential, in terms of their education, but also their ability to form and sustain meaningful relationships, to have success in multiple regions of their lives. And so how do we start to understand the impact of early adversity on the infant brain? Well, most people would say, well, let's look inside the brain, let's do some brain imaging. That seems to be the most direct way of seeing the consequences of all of these different risk factors. Now, the image that I'm showing you here, many of you will be familiar with this technology, magnetic resonance imaging, and it requires scanners like these. Many of you may have been in these scanners. They're everywhere now in high-resource settings. Now, it is possible to get infants into these scanners, and there's a large program of work ongoing at many centres now worldwide, collating MRI data on infant brains. But it's very difficult to find these sorts of scanners in the type of resource-poor settings where the infants at highest risk are growing up. And so there's a clear unmet need here, and the unmet need is for robust and objective measures that enable us to understand the infant brain from birth in resource-poor settings. And so, as a medical physicist and biomedical engineer, that's the unmet need that I've been addressing with this project, Brain Imaging for Global Health. And I'm going to first of all tell you a little bit about the technology that we're using to address that need, and then essentially tell you the story of how this project has evolved. And my disclaimer right up front is that I have no background in global health. Before I started this project, I'd never been to Africa. And I have no background in the type of child development issues that these infants are experiencing. So this has been a huge learning curve, not just in terms of the development of the project, but also for me personally. So the technology that I'm going to talk to you about is called near-infrared spectroscopy. And these are some photos of some infants that we have been scanning here, just a few hundred metres away over at Baby Lab at Birkbeck, just across the road. And this technology was first devised actually by Dave Delpy and others in the 1980s. And it was first demonstrated, its use was first demonstrated on young infants. And now we have a huge programme of work that enables us to study infant brain development for a range of different conditions. So let me tell you how this technology works. Well, it essentially depends on the absorption of light. This slide shows you two tubes of blood. One tube of blood has lots of oxygen in it, so it appears bright red. The other is more of a purple-bluey colour. And if I'm giving this talk to schools, I often get them to visualise the difference between Manchester United and West Ham, and that usually works. But why are we seeing these colour differences? Well, what you're seeing here when you look at this slide is white light falling on this slide. All of the colours, apart from the red, are being absorbed, and the red light here is being reflected back to your eye. So we don't actually need to be able to see an object to know the colour of it. We just need to know the colour of the light that it reflects back. So we can do that with biological tissue. So let's take a finger. If we shine white light into your finger, and you can do this using the flashlight on your iPhone, you'll see that the colour comes out. The colour that comes out is red. And that's because your tissue contains red oxygenated blood. So just by shining light through tissue, looking at the colour that returns, we can get a measure of the amount of oxygen that's being carried in that tissue. But as you can see, here's my red laser pointer here. If I shine this through my finger, it works pretty well, and your flashlight of your iPhone will work equally well across your finger. But if I try and shine this across my head, because I'm interested in looking at brains, it's not going to work so well. So we have to innovate some new physics and engineering, and this is to move from visible wavelengths into the near-infrared, and to use wavelengths that are equivalent to those that come out the TV remote control. They're invisible to the naked eye, but they have a particular interaction with biological tissue that enables us to do this. This is a hand transilluminated with near-infrared light. And what you can't see on this image is the bones, because the light passes straight through the bones. What you can just start to pick out, though, is some of the vascular structure. So what this tells us is that if you use near-infrared light, it will pass through the skull into the brain, and then you can just look at the colour differences in the near-infrared rather than the visible, and this enables us to look at the colour differences in the blood in the brain, and therefore how much oxygen is being carried in the brain. So it's a very specific way of looking at tissue. The other thing that's important to understand about this technology is it's completely safe. It doesn't use ionising radiation. It's completely safe to use for multiple hours on the most vulnerable patients, small babies. And so it has particular applications for a range of studies where conventional imaging, like MRI, may not be appropriate. So the instrumentation is pretty straightforward. There are some people that have said I've spent most of my career working on a glorified torch, which is probably true, but the low-tech technology gets you a lot of places. And so the technology works by firing light from the near-infrared into the tissue of interest. Now, the tissue of interest may be, for example, an infant's head. And all I've spoken to you about so far is the effects of absorption and the colour differences that we can see. But actually, the most dominant feature of light transport in tissue is scattering. And I showed you that earlier, just when I shined the laser light through my finger, and you would see it also with your flashlight on your phone. My collimated beam here doesn't stay collimated or straight in a straight line for very long. It becomes very diffusely scattered. And so when we're passing light into something like an infant's head, we get very diffuse scattering. And so we're actually only going to see a very small number of photons that survive that journey across the tissue. So it is rather more complicated than the glorified torch. We need to think really carefully about the light detection systems that we use and to make sure they're optimized to look at these very low light levels and to discriminate the different colors or wavelengths of the light that are coming back. And so the light travels across the tissue, and we can use that to work out the light that's been absorbed, and therefore the color of the light that comes back, and the relative amount of oxygenated and deoxygenated blood in the tissue of interest. Now, Dave may recognize this photo. This is a photo of an infant on the intensive care unit, the neonatal intensive care unit here at UCH, probably taken in the mid-1980s. This infant's undergoing full life support, but the probes that you can see here are from the near-infrared spectroscopy system, which are providing a continuous monitor of the oxygen levels in this baby's brain, and that's to aid clinical management of this infant. And so you can see how well it fits into a high-tech environment, because it's out of the business end of keeping this baby alive, and it's a completely non-invasive way of studying what's going on inside this infant's brain. So from its first application in the preterm or neonate, very young infants, we've now shown demonstration of its use in a whole range of different applications across the entire lifespan, all the way through from the fetus up to the adult. And I think it's fair to say I could give you a talk on any number of these, but I'm going to focus specifically on the idea of how we can use this in resource-poor settings. But let's think a little bit first about what we're measuring. So I showed this image earlier. This is very recognizable to you, I'm sure, as an image of the brain, because you recognize the structure of the brain. So this is an image of brain structure. Now, we don't typically use our optical imaging to get an image of structure. Instead, we're looking at an image of function. So the contrast that you're seeing on this image here, the different colors here, are showing you where the oxygen is in the brain. And so this is telling us about how the brain is utilizing oxygen, and therefore telling us about brain function specifically. And in fact, if I was to image all of your brains now, I would hope to see that this is what it looks like, because hopefully you're being activated both visually and orally, and your brain is diverting oxygen to different parts of your brain as you're processing the information that I'm giving you. So these are the types of images that we can get with near-infrared spectroscopy. But I want to think a little bit more now about specifically what happens when the brain is activated. You're looking at me now, you're looking at the screen. So let's just imagine what's going on in the back of your brain. If you feel the bump at the back of your head, that overlays the visual cortex, which is responsible for processing visual information. What's going on inside that part of your brain right now? Well, as you're looking at these images, the neurons in that part of your brain are firing. And as they do that, they're using up oxygen. The response to that is to increase oxygen delivery to that region of your brain, specifically the region that's being used to process that information. How do we increase oxygen delivery? Well, we increase blood flow. And with that, we can see changes in the amount of oxygenated blood washing in, the nice bright red blood, but also the washout of the deoxygenated blood. And so if I was to look at the signature, the characteristic signature signal that came from that part of your brain, it would look something like this. So your brain had been activated here. The neuronal activation happens in the course of 50 milliseconds or so, very, very quickly. And the response to that is a very slow, what we call hemodynamic response, where we see the inflow of the oxygenated blood and the washout of the deoxygenated blood. And this hemodynamic response is what we measure with our near-infrared spectroscopy systems. So it's not a direct measure of the neuronal activation, it's a consequence of that, but we can use it as a surrogate measure that your brain is activating and performing normally. So how do we do this in infants? Lots of work has gone on in our department in developing systems that enable us to place multiple sources and detectors, optical sources and detectors onto an infant's head. This is an example of a 4-month infant. These infants are particularly challenging because if they don't like having something on their head, they've got the capability of taking it off. So there's a lot of design, and one of the most important features of this design is the chin strap. And this work was done in collaboration with the Centre for Brain and Cognitive Development at Birkbeck, I mentioned earlier, the Baby Lab. So it was very much a multidisciplinary project with psychologists who are used to doing infant studies, working with bioengineers to get the right design that's going to work in that context. And so using those sorts of systems, we can measure the hemodynamic response that I just described to you. And because we're measuring it over multiple sources and detectors, multiple channels, then we can recreate the sorts of images that you can see here. And the image here again is just showing you this focal point here would be the changes in oxygenation related to the brain function. So how do we find out about infant brain function by looking at these types of images? Well, this is an example of some of the studies that are going on in Baby Lab. So we have an infant sitting comfortably, in this case on his mum's lap, looking in this case at both social and non-social images. And he may also be listening to some auditory stimuli as well. And I'm just going to show you now a video of us filming that infant's behaviour while he's looking at these images. So what's happening here is the infant sat on their mum's lap, and the probes from our near-infrared spectroscopy system, this is an older design, hence the sort of rather ropey-looking bandage, is wrapped around the baby's head. And the baby's presented with a range of different images. Now the baby's looking at non-social images, trains, planes, tractors, not particularly engaged, actually looking at their blanket more than looking at the screen. But in a moment, an actress is going to come on the screen here and do Itsy Bitsy Spider. Here she is. And you can see that infant's behaviour change. You can see that infant engaging with what's going on the screen. Now, behavioural psychologists have known for decades that this is the way that babies respond to social rather than non-social cues. The question they posed to us was which region of the brain is responsible for them identifying a face rather than a tractor. And so what we were able to do, and this work was led by Sarah Lloyd-Fox, who was my PhD student, is now a postdoc researcher and neurodevelopmental psychologist. And what she did was study a range of infants and look for this hemodynamic response and showed that it appeared for the social in preference to the non-social image. Because we were measuring over multiple regions, we could create these maps of brain function. And what you're seeing here is the evolution of a response in a region of the brain known as the superior temporal sulcus. And it's actually the region of the brain, your brain, that you're using right now to interpret me as human. My biological movements, hopefully I'm coming across to you not as a robot but as a human. And we were able to show with this study that this region of the brain is mature from as young as 4 months of age. Why is that interesting? Well, it's interesting because the group at Birkbeck were interested in looking at infants who don't develop typically. And their particular interest is looking at autism. So Sarah repeated this study in 2 groups of infants. In the first group of infants, these are infants with a low risk of autism. And she showed the typical response I've shown you, an increase in oxygenation in response to the social visual stimulus. She then repeated the study in a group of infants at high risk of autism, and the risk factor here was a sibling with autism. So all of these infants had a sibling with autism, but all of the studies, the low and the high risk group, were studied between the ages of 4 and 6 months. And this is a long time before the clinical diagnosis of autism is performed, which is typically in the 2nd or 3rd year of life. So these infants are at risk, but we don't know if they will go on to develop autism. But when she looked at the brain responses of these infants to identical stimuli that she presented to the low-risk group, we see a hugely moderated response in this high-risk group as young as 4 to 6 months of age. This work was published back in 2013, and obviously since then the infants have aged, and they've aged into the point where they can undergo a clinical assessment for autism. So these are the data that I've just shown you represented in 2 different colours now, but it's the same data. The green are the low-risk group showing the typical response, and the purple are the high-risk group showing the moderated response at 4 to 6 months of age. What we can do now is look at these responses and categorise them in terms of the outcome of a clinical diagnosis of the same infants at age 3. And this is what we see. So the infants that were low risk at 4 to 6 months, none of them were diagnosed as autistic, and those are the ones that had the normal response. The infants that were high risk at 4 to 6 months who weren't diagnosed as autistic still had a positive response at 4 to 6 months. But those infants that were high risk at 4 to 6 months and were diagnosed as autistic at 3 years are the ones with a negative response. So this study is the first to show that functional brain responses measured as early as 6 months of life are associated with autism outcome at age 3. So this is a really exciting example of how this technology can be used to give us an insight into infant brain development much sooner than any of the behavioural or clinical signs may be shown. Now, what's the advantage of that? Well, the advantage is then if we know that this infant is going to go on and develop autism, why wait till 3 years of age? Start interventions as soon as you know. So the idea is understand the brain early, understand when atypical development is happening, and start to intervene. And on the basis of the publications that we had written about this work, we were then contacted by this group, the Medical Research Council International Nutrition Group, and they work out of the London School of Hygiene and Tropical Medicine, which is also just a few hundred metres from here, but also a field station in the Gambia, in the village of Keneba. And the village of Keneba is a very low resource setting. There's no running water, no electricity. But it has housed an MRC field station since the 1940s. Most of the work has been looking at nutritional interventions to try and support the work that's going on in understanding how better to nutritionally intervene and support these infants. So there is a nutritional rehabilitation centre. But the researchers that were working there knew that it wasn't just about understanding the physical growth impact of malnutrition on these infants. They wanted to understand the impact that this malnutrition was having on brain development. This is what they were using when they contacted us to understand infant brain development, which is entirely sensible. They were measuring the circumference of the head. And this actually provides very useful information. So this plot shows you head circumference data from infants in the West Kiang region of the Gambia, where the field station is. The green line at the top, these are Z-scores, reference to the World Health Organization norms. And the green line here is where you would expect to see healthy infants, either on or above this green line. But you can see these Gambian infants that all experience what we call head growth faltering over the first 2.5 years of life. So just by making these simple growth measurements, we can see that these infants' heads are not growing appropriately. But it's an open question as to whether that deficit in head growth is associated with a deficit in infant brain development. And so that was the question that we posed in the project that this collaboration started. Can we use the functional near-infrared spectroscopy technologies, which I've spoken to you about before, that we've already used here in the UK and many other places to look at infants at risk? But can we use it specifically in these settings to look at infants at risk of the type of early adversity that I spoke about earlier? Well, that was our idea, and you can't get anywhere without funding. So we proposed our idea to the Bill and Melinda Gates Foundation. They run a scheme called Grand Challenge Explorations, and we were awarded a phase 1 grant of that scheme. That's $100,000. And the carrot that's quite a long way away and is quite a small carrot when you apply for this grant is there's a phase 2 award, which is $1 million. But at the time that we were awarded our phase 1 grant, I was told that the success rate for phase 2 grants was 0.2%. So that's pretty low, even in terms of research funding. But anyway, we got our phase 1 award. On the day it was transferred to sterling, we got a really bad exchange rate. And we could only build a small system to take out to the Gambia. We had a very limited budget. So this system was built in the Department of Medical Physics and Bioengineering, which is the building right next door to us here. And we simply put it in a box and took it with us as excess luggage out to the Gambia, and then drove with it to the field station, which was about a 3-hour drive, the last part of which was on completely unmade roads. So there was a lot of nervousness in the Land Rover that this kit would not survive the trip. So when we were setting up the following morning, we were given a disused library in the field station. So the field station has its own electrical generator and its water borehole to support not just the research that goes on there, but also a health clinic, which is free for the villagers and the surrounding population. But we were given a room, and it looked very different to the type of room that we were used to doing our studies in. And so we just thought, right, let's set the kit up. So the next few photographs shows what happened next. So within an hour, we had converted this collection of boxes here, which is essentially our brain imaging system, into the setup that you see here. This is Sarah Lloyd-Fox, who's a neurodevelopmental psychologist. This is Maria Papadimitri, who's a fellow medical physicist. And we basically set the kit up exactly as we would do in the UK. In just over an hour, we had our field worker, Sekou, and we were training him already to show him how to put the headgear onto the infants. And then just over 2 hours, we had our first mother and baby come in for a study. And so we thought, well, this is going okay so far. Let's carry on. Let's do a study. So we carried out a study that we knew very well. We did the social, non-social study that I've already spoken to you about. The only cultural adaptation that we did was to refilm the videos with a Gambian rather than UK actor. That was the only thing that we changed. You can see the position of the infant, and the mother is identical. And just a few minutes later, we had finished our first study. So I was just snapping these photos away at the time. I just was quite keen to keep a record of what we were doing. And I was faintly pretty amazed at how smoothly it was going that morning. But I'm very glad I took this particular photo because he looks pretty surprised, and maybe he should be. It turns out this is the first baby to have their brain imaged in Africa. And given how many decades of work had been going on outside of Africa in infant brain imaging, it's really quite ridiculous that it took that long, but all we did was put the kit in a box and take it there. I had been tasked with giving a seminar at 3 o'clock that afternoon to the staff at the field station about what we were going to do on our visit. And so it was absolutely wonderful to have already done a study in the morning. Sarah analysed the data, and the plots that I'm showing you here are just the hemodynamic responses that we measured from this infant. And I just draw your attention to these particular plots. So these relate to the different positions of the sources and detectors on the baby's head. And what reassured me when I saw this data was that we had localized responses. So not every single channel is showing a response. We wouldn't expect it to, because not every region of the brain would be stimulated by the visual stimulus that we gave the infant. So these localized responses are really good indication that the technology is working and we're actually seeing a physiological response that we'd expect to see. The temptation, those scientists among you may recognise this, is just to say, well, we did it, n equals 1, let's go home, and that's great, we proved that it works. We didn't do that, we stayed on and we studied 42 infants in this age group of 4 to 8 months, and we started with this age group because that was the age group that we knew best from our studies in the UK. We then went back, those studies were done in the March, we went back in the August and studied these infants again when they were between 9 and 13 months. And then again in the November when they were 12 to 16 months. And then we also added in time points, cross-sectional time points, studying infants at birth and up to their second birthday. And the aim of this was to cover the first 1,000 days of life because, as I said earlier, that's really the critical period where we have this very rapid brain development happening. And we've made a promise to the Gates Foundation, our funders, that we would test the feasibility of the technology across that whole period. We were able to show age-related responses in these hemodynamic response curves. So for auditory, we saw a change in the magnitude, and for the visual, we saw a change in what we call latency, which is just the timing of the response. And this video just shows you an older infant having the scan done. I show this because there were a few naysayers that said we'd never get this to work in Africa and we'd never get it to work on toddlers, so I quite enjoy showing this video. So this little boy is 22 months old. A mirror is just showing you what he's looking at on the screen here. So again, it's a range of social and non-social stimuli that he's looking at. And we can see not only that he's looking at the screen and engaged, but we can see noise is stimulus. But we're measuring continuously what's going on in his brain. So he enjoys doing the peekaboo in response to what he saw on the screen. So the idea is that we're looking at what's going on in his brain at the same time. Because of the success of that project, I started an initiative called Global FNIRS to encourage the use of this technology in global health projects. And also we wrote up our findings in a paper because we were pretty sure that we had that, as I say, the first brain imaging in Africa. The Gates Foundation were pleased with the work that we'd done, and actually it changed one of the topics that they issued for their funding awards to look specifically at methods that could help identify brain development in a non-invasive and objective way. We've collaborated with groups in Bangladesh who have got the same system that again has been developed in the building next door in medical physics, and using the paradigms that we've developed but now culturally adapted for that population. And so now we have data that's been collected on 3 continents that enables us to look at properly at cross-cultural comparisons of infant brain development. But I mentioned to you the funding. We were not satisfied with just getting phase 1 funding. We really wanted to get the phase 2 funding because we knew that we could continue this work. And this involved me taking a sabbatical, spending a lot of time with global health audiences, meeting researchers, understanding more about what the unmet need was and how we could develop systems that would address that. And part of that was a meeting that I went to at the Gates Foundation. I was presenting a poster, and with zero warning, Bill Gates came to my poster. If you've heard, I'm quite involved in public engagement, and so I use this slide all the time now to say to students and staff, you just never know when you're going to have to explain your research. So I had 7.5 minutes with Bill Gates. He's a tech guy, so he loved the tech. Obviously super intelligent, asked lots of very difficult questions. But a combination, I'm hopeful it was partly this conversation, but a site visit that the foundation did here where they were able to see the co-location of all of these different laboratories, physics, psychology, the London School, and also a lot of hard work by the entire team. We've now been awarded over $3.5 million for this follow-up project, which is the BRIGHT project. So it's taken me quite a long time to get the title of my talk. This is an ongoing project, and our ambition with this project is to develop brain function for age curves in both Gambian and UK infants. Many of you who have children will be familiar with growth curves that track the trajectory of your child's growth, but the equivalent for brain function doesn't exist, and so that's what we want to develop. Specifically, we want to look at the trajectories of both typical and atypical developments. We want to identify early biomarkers of disrupted development, and then crucially, we want to inform effective interventions to improve long-term outcome, essentially to provide the infants in these low-resource settings with the potential that they deserve. It's a complicated study. We've added in many more measures than just brain imaging. We recruit the infants antenatally. We measure them up to their 2nd birthday. We start with home visits, understanding the home environment. We do neonatal behavioural assessments. We measure language, caregiving practices, as well as doing FNIRS. We're also doing EEG, which is another form of brain imaging. We do multiple brain behavioural measures, looking at infants' attention with eye tracking, parent-child interaction. We do questionnaires for the health, paternal and maternal mental health, and of course the all-important growth measures, which are a mainstay of lots of the global health projects. We've recruited infants in both the UK and the Gambia. As I said, we have 225 infants recruited in the Gambia and 60 in the UK. So for a global health study, those numbers are pretty small, in fact very small, but for a brain imaging study, they're huge, particularly in infants. This is a photograph of one of our laboratories in Kenneba, in the bright sunshine, which is great, and this is what it looks like in the rainy season, and this is when you realise there's a hole in the roof, so the team had to scramble to move everything. So there have been all sorts of challenges with setting up this project, and because I'm not from global health, it's been a particular challenge for me in working across 2 continents. We've been really invested in capacity building, okay? So the team that's working in the Gambia, with the exception of one UK national that lives there, are completely Gambian. So the science is being run and in part developed by the Gambian team, and that's really important. My ambition really is to create a centre of excellence in the Gambia. This group of African scientists know more about infant brain imaging probably than anyone else on the continent, and I'd like to build on that and ultimately to have them train other African scientists. The results are coming in, and I don't have a big reveal in this talk about what we've found because we're still collecting data. We won't finish collecting data in the Gambia until the summer of next year. But what I can show you is that we are producing data that no one's seen before. So These are some of the brain images that have been reconstructed by my PhD student, Liam Collins-Jones, in collaboration with Jem Hebden and Rob Cooper, who work in medical physics, who've got lots of expertise in image reconstruction. So these are longitudinal brain images of function in the Gambian infants. And this is just a snapshot, which I almost hesitate to put up there, but this is just to give you an idea of the type of curves that we want to develop. This is a paper that's just been published, actually. It's now published rather than in press. And it just shows the responses to a habituation study that we did in both UK and Gambian infants across 5, 8 and 12 months of age. Now, this is just not indicative of our final findings by any stretch at all, but it's showing you that we are generating data. The quality of the data is good enough for us to start to generate these sorts of brain function for age curves. In terms of the field of global FNIRS, which we established, I'm really encouraged to see so many other near-infrared spectroscopy groups take up this challenge. And there are now multiple projects embedding near-infrared spectroscopy in global health work all over the world. And particularly exciting, some of this work was reported at our FNIRS meeting. As you heard, I'm president of that society. We had our meeting in Tokyo in October. And this really it heartens me that we're now seeing the evolution of mobile brain imaging laboratories which can be taken out into resource-poor settings and set up within a matter of less than an hour. And these studies can be done on infants and children of all ages, where previously there was no way that you would have been able to access brain imaging in these populations. Just finally to say that I started with engineering and addressing an unmet need, and that's where I'm going to finish. For those of you that are local to this area, you may sometimes go to the farmers market on a Thursday, which is between here and Birkbeck, and you'll see these hoardings and you might be slightly inconvenienced by them, but they're covering up the building of the world's first toddler lab. So this is obviously the evolution of Baby Lab. And for this, we are now set another challenge. How do you measure brain imaging? How do you image the brain of toddlers that are now going to wander around their environment? They're not going to sit nicely on their parents' lap in front of a screen. The toddler lab is going to embed with funding we have from the Wellcome, a virtual reality cave system, fully immersive system. And so this has now prompted, partly this and other projects has prompted the evolution of wearable FNIRS. So wherever the baby goes, the brain imaging goes with it. So this is what keeps engineers excited, the innovation, the challenge of addressing unmet need continually. So my next ambition is to get a solar-powered, wearable, portable, lightweight device to go on the back of this motorbike, so that instead of this field assistant going out to the villages maybe an hour away and then arranging transport to bring the mothers and babies back, they don't come back on the motorbike, they come back in the Land Rover, is to actually put the brain image on the back of that bike. So we don't have to have the infants come to us. So that's the ambition for the future. I'm going to finish with what's the most important slide, which is to thank the team that have enabled this work to happen. It's a multidisciplinary team, and we use that word a lot, but it truly is in this case in terms of medical physics, biomedical engineering, international nutrition, maternal health, child development, and everyone working together. And I should say there's been a huge number of people working on this project that have volunteered as well. So I'm hugely grateful to them and the funders. Thank you very much.
Thank you very much, Claire. That was absolutely fascinating. I've seen some of those slides, but not the whole story. It's fantastic. We've got 10 minutes if there are any questions that the audience want to ask. We've got 2 people with microphones, so if you could just wait a second for the microphone to arrive, then you're going to be recorded.
Thank you for such an interesting and engaging presentation.
Thank you.
Out of curiosity, with the Gates Foundation, is there any particular reason why there's such a low success rate in applying for the larger funding? That's one question. And is all of this work solely dependent on these type of research and grants and so on that you need to get in order to do the work?
So I'll answer the second question first, if I may, which is absolutely. So I tell the story of this grant in the way that I do because I think it's important to show the importance of how we can engage funders, right? So in the case of the Gates Foundation, it's quite an unusual organisation to approach for funding. They're really driven by the idea of your research, and the form that I filled in was 2 pages, and it was completely anonymised. They had no knowledge of our track record in this field at all. And I would compare that to a research council, which is typically where most of us would get our funding from, either the Medical Research Council or Engineering and Physical Sciences. So those forms are now 120 pages, I don't know, a huge amount of paperwork, with lots of complex costings and everything else. So sometimes the evolution of writing the grant application is hugely lengthy. The Gates is a much quicker process, and so it's all about engaging them with the idea. Why is the success rate? I didn't ask Bill Gates that, I should have done. I think one of the things about the Gates Foundation is that it's very reactive, and I think it needs to be. So during the lifetime of this grant, Zika occurred in terms of the issue with that. So they want to be able to divert funding to make a difference on the ground as soon as possible. Their tagline is they're impatient optimists. So, and they want to have impact at scale. So you know that when you engage with them, that the other thing I was told about them, which I think is true, is they sort of have attention deficit. So you do need to keep on their radar. You need to convince them that your work is important, and you need to be able to keep showing them the impact of your work. Or in our case, it was telling them what the potential impact, once we prove feasibility, what the potential impact could be. But they're a very interesting organisation to work with.
Thanks. Another question? Thank you.
Thank you for your presentation. My question is it difficult to use NIRS because we have to avoid the bias by the experimental or methodological bias? So is it difficult to use NIRS?
Because of the experimental, sorry, what we...
in the experimental method, or?
So the practicalities of using it?
I think it's the question of the degree of objectivity that this might give, as opposed to many of the previous measures, which are, appear more subjective than objective measures.
So, okay, so one thing I didn't focus on, which is maybe worth just reflecting on, is that the best way to understand infant brain development is to see what an infant can do, right? Is to measure whether they're capable of certain behaviours at certain ages. But by default, one has to wait until the infant grows to a point where that behaviour is observable. So I think those sorts of methods are subjective to some extent. What mood is the baby in when you check whether they can climb stairs or pick up a bean off the table? What we're doing with the brain imaging is trying to provide an objective measure where we cut through a lot of the experimental issues that you might have with some of the other type of assessments. The one thing I always say is you can't fake your brain responses. If I was to measure your brain now, we'd have a reasonably good idea of which part of your brain is being activated. Okay, so in terms of an experimental paradigm, the one thing we do have to consider with the infant studies is keeping them engaged with what we actually want to measure. So I only showed you a brief clip of that video. Some of the studies that we have involve assessing these babies for maybe half an hour. But a lot of that is attention keeping of the babies, and it's something that we know is going to grab their attention and keep them engaged. Not that we want to measure the responses to that particular part of the stimulus, but we need them to keep looking at the screen until we present something that we do want to measure. So this is another example, I think, of, as medical physicists, I couldn't design the paradigms where my systems are being used. That's why I have to work very closely with the psychologists. They need to understand the limitations of the technology, which regions of the brain we can penetrate, what type of spatial resolution we can get with our systems. And I need to understand the paradigms when they say to me, you can't repeat the same stimulus 10 times, the baby will get bored and switch off. So it's all about understanding where the limits of the technology and the paradigms are.
Thank you.
Another question?
Just for interest, when you were showing the infants pictures of adults and their actions, Did you ever try any animals? Cats and dogs, mice or rats?
Yeah, the slightly flippant answer to that is it's quite hard to get a cat to do peekaboo. Sorry, but I couldn't resist that. Okay, I'm expanding on that story. So when we first landed in Africa, we literally had the stimulus that I showed you, which was the moving stimulus with the refilmed Gambian actor, where it was actually a student from the London school that we brought over to film. The still images that I showed you previously, like the tractors, we kept those the same. When we got to the Gambia, we immediately realised that those images, the non-human images, were entirely culturally inappropriate, because those sorts of things are not wandering around the villages where these infants are growing up. And so what we did do is go out and take some photographs of still non-human images that we thought were more adaptable or more relevant to that population. And we did take some pictures of animals. And then we had a whole debate about, well, we can't use animals because they might look too much like humans. So we haven't looked at the non-adult effects. What we are looking at now is live studies, where instead of relying on films of actors doing things, we can actually present infants with real scenarios in front of them. They are still adult-based at the moment, but a slide that I actually took out of this presentation, maybe I should have kept in, was a study that's going on in Budapest, where one system is imaging 2 infant brains simultaneously. So then we can start to look at the interaction between infants, which is getting much more realistic about how infants evolve their social interactions, because it's not just with adults, it's also with other infants. So I would say that, watch this space. The wearable technologies are going to enable us to provide much more naturalistic imaging of infants in a whole range of different settings, and understand more about the diversity of their responses.
I think we have to stop there because there's a lecture in 4 minutes' time. But could I just ask you all to thank Clare for a fantastic lecture?